Welcome back to The Explainer. Today, we are setting the stage for what is honestly a microscopic cellular detective story. We're diving headfirst into the fascinating, incredibly delicate world of immunohistochemistry. It's this high-stakes realm where lab technicians are basically acting as investigators, hunting for tiny molecular clues that will ultimately dictate a patient's entire diagnosis and treatment plan. But here's the catch. These clues, they aren't just sitting there out in the open waiting to be found. They are hidden, completely masked, locked away. And to find them, we literally have to unlock the tissue itself. Okay, let's dive into this. Imagine you are looking for a highly specific protein inside a tissue sample. It's a tiny, invisible molecular clue that tells you, definitively, if a cell is healthy or diseased. But the tissue has been preserved in a way that literally builds a chemical fortress around that exact clue. So how do we break down that fortress without destroying the cell itself? Well, that is the absolute core challenge of immunohistochemistry, or IHC. It is this incredibly precise, high-wire balancing act of chemistry, temperature, and timing. And trust me, the environment where all this happens, it is far from simple. The typical histology workflow is chaotic. I mean, you practically need 8 arms just to keep up. You've got a multi-armed operator juggling this endless, relentless stream of different consumables, wildly complex instruments, and shifting physical properties. You're dealing with reagents that vary in their chemical makeup, dyes that differ in size and color, all while a literal clock is ticking down. It is an intensely demanding environment where just one tiny slip-up can derail the entire shebang. Now, one of the biggest bottlenecks in this whole workflow is time. Standard histology, the traditional way of preparing tissue, that can be a grueling 16-hour marathon of processing and embedding. But in a medical scenario where a surgeon is waiting on a prompt diagnosis, waiting overnight simply is not an option. Enter frozen sectioning. By taking water-rich tissues and freezing them rapidly with a microtome cryostat, an operator can cut sections and have them fully ready to go in just 5 minutes. Boom, 5 minutes. This rapid freezing stops the tissue from disintegrating, and crucially for our detective work, it preserves the chemical makeup of the cells without hiding it, which is exactly why these cryosections are so heavily utilized in immunohistochemistry. But what if you aren't using frozen sections? Because the reality is, most tissues in pathology are actually preserved using formalin fixation. Now, formalin is kind of the necessary villain of our story. We absolutely need it because it preserves the tissue morphology, right? It keeps the cells actually looking like cells and stops them from degrading. But there is a massive catch. Formalin fixation creates structural crosslinks, specifically these things called methylene bridges, right across the proteins. Think of it as throwing heavy microscopic chemical handcuffs, or this dense impenetrable net over our target clues, which scientists call antigens. Because of this net, the antibodies we use to detect those antigens, they just bounce right off. The targets are completely masked. So, how do we get past the mask? Well, we use a technique called antigen retrieval. Let's actually take a second to really focus on this, because it's absolutely crucial. Antigen retrieval is our primary technological solution to that formalin problem. It's a highly deliberate, carefully calibrated method designed specifically to break those methylene crosslinks. By dismantling that chemical net, we reverse the masking effect, unfolding the target sites so that our diagnostic antibodies can finally actually bind to them effectively. We generally have 2 main weapons in our arsenal to pull off this unmasking. The first is heat-induced epitope retrieval, or HIER. HIER uses really high temperatures combined with specific chemical buffers to quite literally unfold the proteins and snap those crosslinks. The second method is enzymatic retrieval. So instead of heat, this method uses specific enzymes, like proteases, to act like microscopic biological scissors, literally digesting the masking proteins to reveal the antigen hiding underneath. Let's ground all this theory in some actual physical lab actions. Say an operator chooses that enzymatic route. The protocol is highly specific. First, they have to deparaffinize and rehydrate the tissue sections. Next, they must place the slides in warm water. Now, why is that so critical? Well, think about it. Placing cold slides directly into a warm enzyme solution will instantly drop the temperature of that entire bath. This immediately reduces the enzyme's activity, which leads to under-retrieval. Basically, the chemical mask just stays on. Once properly warmed, though, the slides sit in the enzyme for 10 to 20 minutes with some gentle agitation, before a crucial final wash in running tap water to completely halt the reaction. Now, while enzymatic retrieval is fantastic for certain difficult tissues, heat-induced retrieval, HIR, is actually preferred for most antigens. Why? Because the conditions are much easier to control. But listen, controlled for sure doesn't mean easy. To optimize HIR, lab techs will literally use domestic stainless steel pressure cookers, scientific microwaves, or even vegetable steamers. And finding that sweet spot requires rigorous empirical testing. Operators have to test multiple retrieval intervals, say 1, 2, 3, 4, and 5 minutes, just to see what works best. They meticulously adjust the pH and experiment with different chemical buffers like tris, ADTA, or citrate, trying to find that perfect environment. And above all else, they absolutely cannot let the slide dry out. If they do, they'll end up with irreversible, patchy staining artifacts, ruining the whole thing. And this brilliantly illustrates what happens when all that hard work pays off in a highly readable physical reality. We get these crisp, beautifully colorful tissue slides where the cellular structures are perfectly intact and those specific target proteins are brilliantly highlighted. I mean, it's gorgeous. This is the ultimate canvas that a pathologist needs to actually make an accurate diagnosis. But wait, unmasking the antigen is really only half the battle. Mastering the final staining steps is just as delicate. Before they even stain, slides have to be drained vertically and dried at exactly 60 to 70 degrees Celsius. Push that temperature even a little bit higher, your precious tissue section might literally crack right down the middle. The operator also has to be a super keen observer. If they notice muddy hematoxylin or pale eosin, those are the classic dyes that give the cells their vivid blue and pink colors. It's a giant red flag. It means the dyes are deteriorating and need immediate replacement. And whether it's washing the section in running tap water for exactly 5 minutes until it turns blue or using a specific weak acid differentiator, the golden rule always remains: do not alter validated staining times. So the crucial point is that troubleshooting this entire process is a true artisan's balancing act. It's like walking a tightrope. If the operator plays it a little too safe and under-retrieves, well, the epitopes remain masked, resulting in weak or just totally absent staining. But if they push too hard and over-retrieve, they get this chaotic background noise of non-specific antibodies binding everywhere. If they use excessive heat in that pressure cooker, the tissue simply lifts up and washes right off the slide. And if they leave it in the enzyme too long, those proteases will cause severe morphological damage, basically destroying the exact cell structures they were trying to observe in the first place. Navigating all of these pitfalls requires immense skill. As our source material so wisely notes, an educated operator will minimize repeated work, decrease time and laboratory costs, improving performance in patient care. I really want us to pause and highlight those last two words, patient care. We can get so caught up in the fascinating chemistry of the buffers and the proteases and breaking those methylene bridges that it is super easy to forget the real stakes here. This isn't just some abstract chemistry experiment. A perfectly retrieved, beautifully stained slide is the direct link to a proper medical diagnosis and a life-saving treatment plan. So next time you hear about a medical miracle, definitely spare a thought for the unseen lab workers juggling all these variables in the histology workflow, wielding heat and enzymes just to uncover the invisible. It kind of leaves us with a profound thought. What hidden cellular secrets could your next slide reveal if you hold the precise key to unmasking them? Thank you so much for joining me for this explainer. Keep wondering, keep learning, and I'll catch you in the next one.